we're setting a standard for spatial biology, and it's really about this idea of differentiated quantitative phenotyping. We have a system that allows you to reliably multiplex, very reliably multiplex, because of a chemistry that's very simple and very clean. We also have a system that has best-in-class resolution of 180 nanometer per pixel, and as well as something that we do that's called HDR, high dynamic range imaging. So to us, this is a standard in optically based spatial biology that we're very proud of. So I'm going to talk through some of the features of this technology as I go along here. So the way our system works is this is a basic summary of the workflow. It's an automated and closed system. You take a tissue sample, fresh frozen or FFPE, you load it onto a chamber or a slide, it becomes a microfluidic chamber, and you do your antibody staining on the system. So you don't stain on the bench, you don't do it by hand, it happens on the stainer. Then you take an image of it, and then we use photo bleaching to remove the signal. And it's an iterative process, it repeats itself. So you can choose your plex, you can use as many antibodies as you need to answer the questions that you have. So it's relatively straightforward. We're going to touch on 3 topics here. One is the performance of the system around sort of the parameters, the optical parameters of it. We're going to talk about the technique, the assay technique, and how we design our assays. So and those are the topics that I'll touch on here. So let's talk about resolution and dynamic range first. The question is, why does differentiated quantitative performance matter? Well, I think anyone here who analyzes data will say because it's probably the single most important thing about the data that we create. They have to be clean. You have to have excellent resolution. You have to be able to see, cells ideally with substructural details. So segmentation algorithms, they rely on us taking the best pictures possible. This is something that we can guarantee with this system. We have extremely good resolution. You can see that with these 4 little grayscale images there. The one on the very left is an image that was taken on our system. It is a much crisper plasma membrane. It's easy to differentiate even in adjacent cells. Someone who uses an unbiased clustering algorithm to analyze their data, they will appreciate this because the output will ultimately be much cleaner. The other feature about this microscope is what's called high dynamic range imaging. Proteins and other biomarkers have extremely wide dynamic ranges or expression ranges within a tissue. And what you can see are a series of 4 different exposures, each capturing the same scene. Each exposure will capture something very different. Now, if you look at the yellow square, you can see a cell on the top left. Basically, at the 87 millisecond exposure, you don't see much. You start to see that capture as you progressively increase your exposure time. But then again, you don't want to overexpose your tissue. So what high dynamic range imaging is, it is a combination of multiple exposures that extends the range of your camera. It extends your ability to capture a biological signal and therefore maximize what you collect from your tissue. That's what really sets the system apart, and that's what ensures optimal quantification of your data. Our system does that by default, so it is part of the process. Now what really, when we talk about setting the standard, thus far, we worked on a custom slide design, so it required a different cover slip to do this experiment. But we're proud to introduce and announce this last Thursday, the CellScape whole slide imaging chamber. You can see here, we now have a microfluidic chamber that makes any standard 1x3 inch slide that you find in cores or histology labs around the world compatible with our system. So to this end, we have a little video that we made here that I'll play for you. But here you can see the slide. Everyone has seen a slide like this. We have an assembly tool. You can load your slide in there. You clamp it down. And your slide with your tissue on it has now become a microfluidic chamber. You can load this onto our microscope. Use a slot for 4 of these at once. So you can do 4 of them. You can image them different formats. There's a TMA on there. There are whole tissues. And when you maintain that top-class resolution and HDR imaging that I had just talked about it. This is a plug-and-play system. You close the hood, you walk away, and you let the data acquire. This is what we come to expect, and this is what we're happy to deliver with. So doing this on a whole slide, I'd like to emphasize this is the largest imaging area with 16 on 40 millimeters. None of the detail or the resolution that I had alluded to earlier, again, has been compromised here. In fact, imaging data that we see nowadays are probably even better than what we had with our previous design. I would also like to mention that by changing the fluidic chamber. If there are any of our existing customers here, we've been able to reduce the reagent volume in this compartment significantly. So you incur, yeah, I would say a noticeable amount of savings in your reagent throughput here, because you need less solution to stain your tissues. And it is an important factor, especially for people who do experiments in the 20, 30, sometimes 50-plex range. Antibodies aren't cheap, so you don't want to waste reagent. This is designed to take that into account. And then here's just an image that we've taken recently just to sort of re-emphasize the point that this is really good optical performance for an epifluorescence microscope. Here you can see an FFPE human tonsil that's stained with some standard immune oncology profiling biomarkers. I can see here in this little zoom-in image, there are two little blue arrows. And what these two little blue arrows are pointing at, if you look closely, those are split nuclei. You can especially see that on the top one. You can see DAPI has essentially, this cell has just undergone mitosis. So this is an observation that you just make on this scope, because really the image quality is very, very good. So these are B cells in a tonsil in a proliferating region. And this type of morphological detail, together with really nice biomarker staining, allows you to not just ask new questions, but to validate your findings through a bunch of different proxies or parameters. So that's kind of what that sort of describes our optical imaging and what our system can do in terms of acquiring data. Moving over to assays, the other very important part about spatial biology. You take a picture, but you need something to take a picture of. To this end, we're very happy to have introduced in the past, but once again now, our VistaPlex kits. VistaPlex kits are antibody assays that are really tailor-made or designed for immune oncology. They're meant to be small enough that you don't need to overcommit to a panel, but the content has to be right so you can ask the basic questions. This is very important. Once again, you're going to spend a lot of money on assays. We want to make sure that you get an assay that answers your questions and it doesn't do anything that you don't want it to do. So what enables us to build these VistaPlex panels, I would say easily and relatively fast, is that we actually exploit direct immunofluorescence as our primary means of staining tissues. In direct immunofluorescence, you have an antibody that carries a fluorophore already. Many of you may already be using these antibodies in histology applications or flow. These are widely available. They're made by the antibody suppliers. And especially for immunology and immuno-oncology, there are many targets you can pursue readily out of a catalog from the big antibody houses. We built our assay to recognize these antibodies, and you can use them. So having these antibodies ready to use, it's easy to build an assay. They also have advantages over other strategies. We don't need to attach an oligo for our antibody. Sometimes attaching an oligo is problematic, changes the affinity of an antibody, something we would like to avoid. We don't strip in our tissues. We don't use low pH. We don't use other or high temperature to remove antibodies. So we try to also, or by that means, we don't really damage our tissues. So we think that direct eye immunofluorescence is probably one of the mildest ways to go about cyclic immunofluorescence. You don't damage your tissue. You don't damage your antibody. So you should be really recording the tissue or the biomarker in its most natural state. I would argue that that's what we all want as scientists. So to that end, using that strategy, our goal is by the end of this year to build a comprehensive set of panels or modules, if you want to call them that allow you to go into the tumor microenvironment, starting with immune profiling, looking for the lineages and some checkpoint markers. This kit is already available. It's been released, I think, at the beginning of this year. Then looking at the tissue architecture, and then pinning down individual cell types, like myeloid cells or NK cells. These are all bite-sized modules. They're not really large panels. You're committing to, say, a dozen or 15 antibodies at once. You can mix and match these modules, and you can ask your way through the tissue as you see fit. This here is an exercise to that end. So this is a breast tissue. And what we did here is we stained this breast tissue with our immune profiling kit. Breast tissue is not traditionally known as an immunogenic organ, but you can see here a lot of red cells, CD8 cytotoxic T cells. So the question is, are these really cytotoxic T cells? Is this tissue infiltrated? Our kit contains a number of adjacent markers that would allow you to answer that question. You have CD3, CD45 in green and yellow, as well as other lineages, such as this lonely B cell that you can see here in purple. So with the right combinations of markers, the resolution, and the detail, you can detect any cell type you want, no matter how rare it is. And that is what we aim to provide here. But one really nice feature about this is, once you have immune profile at this section, and you think this is a good experiment to pursue, you now looked at this. Maybe it took you a week to analyze these data. You can take that same section, that same chip, load it back onto the microscope, and you can now reinterrogate it. So what we've done here, we took this section that was labeled with these immune markers. We described the T cells. We had a cup of coffee. We mused about what all this could mean. We put this architecture kit on it. So you can see in yellow and green, we're now looking at the substructure of the epithelium. And we're providing context to the immune infiltrate. So we've taken that very same section 2 weeks later, put a whole new module of antibodies on it, and we've now revealed that there's T cell infiltration in lobular structure, in secretory ducts, but not necessarily in the adipose tissue of this breast. We still have this sample in the refrigerator today. It's possible that we go back to the lab on Monday and we put yet another round of antibodies on that. I think for us who spend a lot of time building antibody panels, this is a very nice feature because, again, you decide when it's time to move on. You decide how much time to invest into the panel that you want to build, and you let the tissue guide you to get there. You're not putting 50 antibodies down onto a tissue that you don't know and end up wasting a whole bunch of money and time. You go along as the tissue tells you is appropriate. That is very valuable, especially valuable for any lab, in the academic sector. So we do really need to be cautious how you invest your time and money. So I personally very, very fond of this feature. Last thing I want to get to for assays is it's very important when you work with antibodies, evidently, to validate them. I think it's important for us just to disclose what we do, how we do it. We're very proud. We spend a lot of time on antibody validation. The first, again, we obtain our antibody content ready to use from suppliers. The first question is obviously, are they labeling their intended targets? So this is a qualitative examination. You take your antibodies, are they labeling the right region, the right cell type? You do co-localization analysis. This is what you see on the top. That's a human tonsil. We're looking at germinal centers and the marginal zones. And yeah, so the staining is what it should be. Your B cells are expressing Ki67, PD-L1 is in the epithelia. All of this has been documented before. We're not inventing it, we're just reconfirming it. But then we go through a lot of trouble, actually, to look at the robustness of the assay and the repeatability. And you can see in the little schematic there on the bottom left, we take we have several blocks in the lab that we know quite well, that we've characterized well. We cut them and we send serial sections to multiple sites to run on multiple systems with multiple operators. So in our particular experiment, there will be 3 different instruments, 3 operators in 2 geographical locations, the United States and Germany repeatedly. And what it shows you on the graph on the right side is how many cells did the operator find on a system in serial sections. And essentially, it's just a number of cells per staining type, per morphotype, or phenotype. It's essentially analogous. So we spend a lot of time doing that. We look for all of the antibodies that we put into a kit. But what this shows you is you get the same result no matter who does the experiment, no matter where it does, at least in our environment. I think that is absolutely crucial because you need to be sure that your assay works. It works as advertised, and it does so over and over again.